Welcome everybody to the Vaccinium CAP webinar series. My name is Amaya Tucha and I'll be the moderator for today's webinar. Today we will be discussing assessing the post-storage texture and appearance characteristics in blueberries, lessons learned and applications in blueberry breeding. We're very fortunately that we have two speakers for today's webinar. First, we'll have Dr. Massimo Aiorizzo, who is the project director for the Vaccinium CAP project and is an associate professor of small fruit and vegetable genetics and genomics in the Department of Horticultural Sciences at the University of North Carolina. His presentation will focus on understanding the relationship between texture and other quality traits at harvest and post-storage. And then we will also have Dr. Lara Jongo. She is the head of the Berry Genetic and Breeding Unit at the Fondazione Edmund Mack in Italy. And her presentation will focus on integrating texture analysis into a blueberry breeding program, how and why. Before we let the speakers start, I want to make sure that everybody stays muted during the presentation so we don't disturb the speakers. And also, if you can please keep your cameras off. If you have any questions, please type them in the chat box or in the Q&A, and we will be asking the questions to the speakers after we're done with the webinar. And with that, I will let Dr. Massimo Aiorizzo start with his presentation.
Maya, can you hear me?
Yes.
Okay. It's in presentation mode.
Looking perfect.
Okay, thank you, Amaya, for the introduction, and everybody for joining the webinar today. Like Amaya said, we will have two-part webinar today. My part will be talking about two recent studies that we completed as part of VacCAP project that aimed at understanding the relationship between texture and other quality trait at harvest and post-harvest and post-storage. So this part of the research is part of the objective one of the VacCAP project, which aim at understanding the link between fruit characteristics and fruit quality characteristics. And the goal here is to identify fruit characteristics that correlate with higher fruit quality and also standardized phenotyping method that can be used by the community. So last webinar, Ilko spoke about assessing the predictability of sensorial texture descriptor using instrumental measure in blueberry. So it was another part of the texture work that we have been doing. And the outcome of that study indicated that there is some texture parameters like maximum force that are best predictor of sensorial texture attribute, like springiness, hardness, and crispness, and other parameters like the young modulus were highly correlated with sensorial attribute. That work was done at the pre-storage or very short storage, and then we have another study that is under preparation where we looked at consumer preference versus texture measurement. And willingness to pay from consumer, we see that increase with increasing maximum force and young modulus. So all those two studies, these two studies are looking at the texture at the prior long-term storage. So today we will focus more on the efforts we have been doing on understanding the relation between texture, appearance, and chemical composition in regard to shelf life, in particular texture loss or firmness loss, weight loss, wrinkle, linkage, and mold. So some studies before have already addressed some of those questions. A few studies described the post-harvest fruit softening. It's related to initial firmness measured with the Firmatech. A few studies related weight loss to stem scar diameter, surface volume, and wax composition. However, there have been very limited study, limitation of some of the study where the number of genotype used in several of those studies were relatively low, from 2 to 9. The texture was evaluated with only one parameter using the Firmatech, which measure a single parameter and simplify the model of assume that the texture is controlled by, contributed by a single parameter. No investigation on shelf life predictability has been done, and the relation to main chemistry trait has been looked at. And then also study across locations experiment were not directly comparable because different method and different experimental design were performed on those studies. So here, what we did for we designed 2 experiments and 2 studies actually. One, a first study that where we looked at 2 large population in Florida and Oregon of 1,000 genotypes each. The material in Florida represents Southern Highbush, the material in Oregon represents the Northern Highbush, and were evaluated for 1 year for multiple parameters. And another study in North Carolina used 61 cultivars representing a mix of North and Southern Highbush and Rhabdi, and evaluated for 2 years. For all those studies, what we did, we evaluated texture using the texture analyzer with 70 collecting 7 parameters, appearance trait like stem scar diameter, berry size, weight, height, and wrinkle degradation, and then chemistry parameter, pH, tartarbol acidity, solute salt content, and sugar at the 24-hour after harvest, and then 6 weeks post-storage. In particular, the chemistry was not done in post-storage, but texture and appearance were done 6 weeks after storage. And these were the storage condition, 3-4 degree, 95% relative humidity in Oregon, and then 1 Celsius in Florida, 95% relative humidity in Florida. And then in North Carolina, 2 Celsius and 80% relative humidity. And in North Carolina, we also added a second time point at 2 weeks post-storage. So again, the experiment in Florida and Oregon was 2 time points, and then North Carolina were 3 time points. So to do this work and to go through all the samples that we have been evaluating, we developed an automated fruit characteristic phenotyping system to evaluate the texture and appearance characteristics. It's basically a texture analyzer that is connected to a computer, and then connected also to a precision scale and a digital caliber. And this, in this way, we could collect automatically all the parameters using a macro, integrating all the traits in 1 single file, collecting the 17 texture parameter and the appearance trait. Texture was evaluating using the texture analyzer, a penetration test with a 2 mm flat probe, and some of the parameters were measured before the break of the skin, and some were measured after the break of the skin. So the study are summarized in 2 papers that have recently, one has been already published and one is in press in post-harvest biology and technology. So what these 2 studies are telling us about texture and the relation to post-storage. So the first thing is we looked at the relationship among all the texture parameters in all 3 studies, and then we noted that we could identify 4 components contributing to blueberry textures. The first component is made of these highly correlated parameters, Young modulus and the F1 millimeter, and which measure the stiffness of resistance to deformation of the berry. The second group, the second parameter were maximum force and FLD, and this is a second component that seems not to be highly correlated with the other parameter. The third component is mean internal firmness or force, which seems to be independent from the Young modulus and other parameter measured here. And the fourth component of highly correlated parameter is represented by DFM, LDFM, burst strain, or FM. And DFM, it's actually represented the deformation before the skin break, or we could also call that sort of ductility, so the resistance to break. Another thing that was noted in all this correlation analysis was that weight and diameter and FLD are highly correlated. And we already knew from previous study that weight and size in blueberry or surface area are representing the same measurement. So you can measure the height of the berry, the weight, or the volume, and you get the size of the berry. The other thing was that chemistry trait like pH, tactable acidity, sugar, and Brix, they are very low or no correlated with texture parameters. So seems to be two independent components. So then here it's the pattern of the texture parameter measured and the appearance parameter measured at the harvest and 6 weeks post storage. You can see that some parameters are changing a lot. The green and the red are the two measurements at the 2 time points. Some parameters increase and some parameters decrease during the storage, and some are stable. Size-related parameters are usually stable during the storage. And we know that the Northern Highbush in Oregon changed more, maybe because the higher storage temperature used in Oregon. So we looked at the relationship between the trait, the measurements of the texture parameter and the appearance parameter at the T0, and which is the 24 hours post harvest and then 6 weeks post storage. And then we see that the correlation is positive. So which means that the selecting for texture parameter at T0 drive the value at the T6. So then we ask the question, can the storage be a factor in separating the data that we collected? And then we see that storage time contributed to separate the data at harvest and post-storage in all set, Oregon, Florida, and North Carolina. We did a couple analysis, PLSDA and PCA analysis to look at this. And then the interesting part was that across the 3 studies, similar parameters contributed to the most of the separation. Probably those were the parameter that changed the most and make the separation between the groups possible. So one of the components it's made of, one of the set of parameters it's made by the Young modulus and the F1 millimeter, which I indicated as being one of the components of the texture. Then the other one is DFM, a separate component. And these were the same component that changed the most and separated the data across all 3 studies. Then we ask the question, can you predict the value of the texture parameter or the appearance, water loss or wrinkle at the T6 post storage using the T0 value? So size-related traits are highly predictive using the T0, the measurement at the T0 24 hours after harvest. Now, all the parameters that were changing had the most change during the storage, like the Young modulus and F1 millimeter, they have a relatively low to moderate predictability. The data were consistent across the different studies. This is the results in the Southern Highbush material. And these are the results in the Northern Highbush material where we saw that the predictability were a little bit higher. And this could be also because of the storage condition being slightly different here. But we go from 0.4 to 0.5 to a moderate predictability for all of these parameters, where the size-related parameters stay the same. Now, in these 2 studies, the Northern and Southern Highbush in Oregon and Florida were evaluated for the absolute value from T0 to T6. Another thing was that the chemistry parameter at T0 that were used for predicting the texture parameter at T6 had a very low importance in predicting the post-storage texture parameter. Again, confirming that chemistry parameters are not contributing much to texture in blueberry. So then the 3rd study in North Carolina, what was here look at that, not the absolute value, the predictability of the absolute value, but look at that predictability of the change during the storage, okay? And then the change of DFM was the most predictive texture parameters, which is one of the components. Prediction, when we add a time point measurement, the T2 time point, the 2x time point on the measurement, you can improve significantly the prediction. And then weight loss and wrinkle were best predicted by the size of the berry at the T0. We also noted in the Oregon and Florida material in the large set that the size affect the water loss and wrinkle. Also stem scar diameter has a very low correlation with water loss. A previous study had indicated there was a correlation, but seems that when you scale up the number of samples, those correlations are not there anymore. The small size berry had a greater water loss, and there was a threshold of couple after 2 grams, the water loss seems to be plateauing, and the smaller size berry have higher water loss and higher fractional percentage of wrinkle. So in conclusion, so what we learned from those 3 experiments is that fruit size, fruit chemistry, and internal ferments have limited contribution to texture change during the storage. 4 texture components contribute to blueberry texture, so which amplify the need of be able to dissect the texture in multiple components using methods for evaluating texture that are able to dissect the texture. External fruit layers contribute more to texture change during the storage. So the Young modulus and the DFM are measured before the breaking of the berry, and measured largely, mainly the external layers of the berry. Post-harvest to texture is related to initial texture, so it's positively correlated. And then here we outlined some possible parameters to use for selecting for improved texture in post-storage. So this will be high value of the first component, the Young modulus F1 millimeter at the T0, higher value of DFM at T0, higher maximum force, which was the best predictor for sensorial hardness, springiness, and crispness, higher mean internal firmness, and then large size, which mean also lower water loss and wrinkle. Now, when we go back to the original pipeline that we developed for measuring this trait, so what we can say now is that you really don't need a caliber, because it seems that, at least for what we evaluated here, that the scar diameter doesn't affect much the post-harvest quality of the berry. You don't need a scale because the texture analyzer collect the height of the berry, so you don't need to weight the berry. And then from 17 parameters, you could go down to 4 parameters for measuring the textures. So here I asked then the question, can you really select for all those parameters together? So, and here I took the Oregon material, which includes 951 genotypes, and then looked at genotypes that have higher than average value for FM and maximum force and Young modulus. FM maximum force is the parameter associated with hardness, springiness, and crispness, as evaluated by sensory analysis. And the Young modulus are the parameters that are changing the most during the storage. And you can see that they are positively correlated, but there is a large number of samples that you could select for if you select only for those 2 parameters. Then after we tried to add another component, DFM, and then these in red are the number of samples that you could start to select for if you want to include these 3 parameters in your selection process. And then here I added the diameter, average diameter of 15 millimeter, and then the number of samples that you can select stay relatively stable with the one where we included DFM. So just to give an idea that if you start to use this method, there is a possibility to select for those traits in your breeding program. With this, I want to just acknowledge all the collaborators that have been involved in this work, group at USDA IRS team at University of Florida, and group here at NC State that have helped with the the chemistry analysis, and then Dr. Lara Giongo, who helped us to set up the texture method. And then acknowledging the scientists that have been directly involved in analyzing and collecting the data, Dr. Marty Pottorff, Molla Fenty, and then Hidu Ko, and of course USDA for funding the project. Some other resources related to shelf life or texture that you can find in our website, the VacCAP trait shelf life that you can look up, the previous webinar about texture in blueberry and cranberry, the barcode system that we developed for processing all the fruit that we had to evaluate, and then the idea is now to try to develop interacting with breeding programs to transfer the method. We have already started to interact with a couple partners that are looking at transferring the method. And I hope that this webinar will trigger more interested on using the method. With this, I thank you for your attention, and will pass the ball to Dr. Lara Giongo.
Thanks a lot, Massimo. Sorry that I just share my screen.
Just to remind, we will take question at the end of Dr. Lara Giongo presentation.
Hope everything is clear with my screen.
Yeah.
Looks good.
Good. So, good morning or good afternoon or good evening to everyone, and thanks for this organization of the webinar. I'm here to present you just my example, so how I developed the blueberry breeding program here at FAM, integrating since the beginning texture analysis. So just kind of a story of our breeding program and some inputs that was validated through all the work that we did with VacCAP project. So when I started, sorry that I need to, when I started the blueberry market and the areas planted were very different. The reality of blueberry was absolutely different than now. So in 2009, 2010, there was still a majority of traditional environments and still quite limited germplasm. Today, we face a lot of different environments than then, and definitely more varieties. Today, it is quite mature market, although in great evolution and highly demanding and challenging, especially for genetics. So just to show you two aspects of, I would say, the same story. So with my unit, my group, we have always focused on the applied research, and this story started in 2013 with a publication on texture analysis on blueberry, because this was a real need since the beginning for selection for us. And then this evolved with VacCAP in a definitely deeper and common public knowledge, validation of the parameters and better application for it. On the other side, of course, definitely integrated with the commercial breeding, we hoped to develop, and the main input was definitely at the beginning the blueberry attraction. So here you can see a visual representation of the breeding program that we developed. And I was quite lucky to start a brand new program, which had some advantage and some disadvantage, I would say, because I started from zero. So I also started, however, with a great generosity from the research community and the industry, and I circulated a survey in order to understand around 2009-2010, in order to understand what were the most important traits to select for. And of course, mainly the industry said yield at that time, but firmness was second for all the categories. So when I started in 2010 to widen the gene pool for Northern Highbush, these were meant to be the main selection traits. Northern Highbush, because we are located in a northern area, and so for me it was the only way that time. Then this developed into a different breeding program in 2014, because I had a chance to enlarge the breeding program in Huelva in Spain. So I was able also to produce and to widen the gene pool of Southern Highbush, and then develop Northern, Southern, and hybrids into new cultivars. So I chose different breeding approaches that are actually quite traditional, but since 2013, so very early in terms of selection, we started to use the texture parameters as biomarkers. After and the same year in 2012, we also developed for texture a mapping population that was Draper by Biloxi. That was a mapping population that was supposed to merge two main traits that we were interested in, texture on one side and flowering pattern on the other side. And then having the chance to have these different pools, I also had the chance to improve a little bit the speed of the program through speed and shuttle breeding. At FEM, we have good platforms for genotyping and phenotyping. So since 2016, more or less, we started to use them as routine. So as I said, it's quite traditional for the breeding approaches. We use conventional breeding, interspecific hybridization, and mutation breeding for the basic work. However, what I chose was the system, let's say, that integrates different strategy to accelerate the genetic gain and new product. So this means that the mantra was to save time and cost, remaining as robust as we could. And the points, the bullets into the green and into the green line are those where we are at the moment, and we were over the time stronger. So what does this mean? That essentially we had to have specific target traits. We had to develop a good relationship with growers that were very supportive with us. We had to develop management-related technology skills, especially soilless technology, because our breeding program is all soilless. And as I said, since the beginning, or very soon, two main divergent environments. Breeding approaches and post-harvest were a key in order to increase the diversity of the germplasm, and post-harvest was one of our priority because of the area we are located in, but because we saw that it was a key also for texture and for quality in general in blueberry. And for what concerns texture, that was our quality-related trait of interest since day one. And this was also confirmed by the good relationship with the market and with the supermarkets in particular in Europe that since more or less the beginning of the breeding program started to test our advanced selection. Something else that I must say related to the breeding priorities, although we can be focused on texture, is how important the selection intensity is, the number of seedlings and crosses per generation in order to accelerate the cycles and the genetic gain and the heritability and complexity of the trait. So these must all be clear. So as I said at the beginning, for us, the question was, can we measure texture and understand the mechanical properties as a function of quality? Of course, there are a few things that have to be underlined. First of all, the germplasm we deal with for answering this, but also to measuring texture, how this texture contributes to quality. Of course, we need to consider the post-harvest as the texture in post-harvest as a component and function of quality, and the diversity, not only of the germplasm, but also of what we are required for by the market. Consumers require different things, so we need to call them. As I said, very soon, in around 2013, 14, texture became a routine analysis to validate selection both at harvest and post-harvest. And what you see in the pictures is related to the publication we validated then within VacCAP 2 years ago with 3 different methods. We use at the moment a different machine than the Text Plus that Massimo showed before. We use the Zwickrol texture analyzer, and but there are different machines that can do let's say the same work. How do we apply the texture analysis into our evaluation? We ordinarily use it for validating the selection that is done in the field, and you can evaluate 50 selections per day, merging the mechanical and acoustic profile, including also the single berry weight and the single berry diameter in the same analysis, as Massimo said. It is confirmed also by the VacCAP results that the 3 main parameters we selected for that are the apparent, what we call the apparent Young modulus, maximal force and deformation, are those more important, but there are also other important for traits correlation. Just to underline how you can increase the process. Ordinarily, you can do, as I said, 50 selections per day, but with an operator as the one we have, Matteo Aielli is definitely more than well trained in our lab to do texture analysis, is able to do very easily about 130 selections per day. So it is also a matter of routine and exercise. Of course, also related to this, we need to think about time and costs and how we can merge within a single phenotyping technique different traits. Going back to what Massimo said, his concluding remarks, we were absolutely happy to see through VacCAP that what we were doing was validated. So the 3 parameters were the same that we are still using for the majority into the texture evaluation. we know that external fluid layers contribute more to the texture change during storage, and that harvest the texture evaluation, the texture profile that is present at harvest is also is mirroring what happens at post-harvest. The parameters, as I said, are those flagged in green, and they are absolutely in agreement with what Massimo said. Relating to the larger size of the berry, I would just indicate to be careful also applying the texture analysis to the carpus anatomical structure that can be really different in different jumbo fluid, for example. So here, what I would like to show you is a bit of evaluation we made into our selection, just to show you how things evolved into the breeding program. So here is a comparison of the three main mechanical parameters: apparent young modulus, maximal force, and deformation. And you can see 71 cultivars versus 240 of our advanced selections, selected, of course, through texture analysis, plotted into the same PCA. And as you can see, we were able to gain genetically for the individual parameters, but also cumulatively speaking for the texture. Here you can see a single parameter plotted versus the year of release when we talk about cultivars, or the year of selection. And as you can see here, we are talking about apparent young modulus. Of course, here our mean young modulus is 2.3 megapascal, is a bit higher than the one that Massimo showed, because it includes all the advanced selections. And as you can see here, you can improve a lot the selection, selecting for the individual parameter. The same can happen for the maximal force. Here is again a plot of about 1,000 genotypes, cultivars and selection. And here is a still higher separation when we introduce another component that is intrinsical into the texture analysis that we have started to work at a couple of years ago, also within the VacCAP project, that is the acoustic component. Until now, we have spoken about mechanical texture, but texture is not only mechanical, it is also acoustic. And as you see, when we analyze the acoustic component merged to the mechanical one, the separation is higher. Another really interesting thing over these blueberry genotypes that we were able to measure over 2 years is that the two components are orthogonal, the one to the other. So the parameters related to the sound, it is not only crunch, it can be other sound that the fluid produces that we want to call in a specific, with a specific adjective, is orthogonal to the mechanical forces. So this tells us that genetically speaking, the control is different. So this is absolutely interesting for the future in order to see what genes are involved in this. So this is these are the results that we had with the commercial breeding program, our first result. And of course, applying texture into the selections means definitely to choose also with other and make a compromise also with other traits into a new product. But all of these starting materials were selected through texture in our breeding program. Of course, relating to the year, because getting from 2015, when we started to have a relative genetic gain to what we have now, of course, the increase is different. So why I believe that texture is a golden trait for quality and to work at in a breeding program. Of course, because you can dissect, you can call, as I said, name in a different way what you select in the field as a subject. And you can give to that subjectivity a correlation with robust data that might be offered to consumers that can ask to the breeders different things. Texture is important, as we said, because it has a strong relationship with post-harvest, and this is a key both for shipping, but also for producing or supplying supermarkets in different windows. It is important for mechanical harvest, of course. It is important because it has a relationship with the development of mods, for example, or resistance or at least tolerance to some insects. And in all these ways, it can reduce waste. As I said in the previous slide, it can also tell us and give us a methodology, an objective methodology, to measure the acoustic and to have new tools to associate in the future with maps that we might produce. So I need to acknowledge for sure my group here at FEM, the VacCAP project, and the whole network that allowed to validate and transfer the method, and all our growers, marketing partners and collaborators, university and stakeholders. And I'm open to accept questions. Thank you.
Well, thank you, Lara and Massimo, for great presentations. Before we start with the questions, I just want to make sure that everybody knows that we will be sending a post-webinar survey. If you can please help us by answering it and providing some feedback so we can improve your experience through these webinars. And also, if you have any suggestions of topics that you're interested in that are part of our project in VacCAP, we would love to hear about that so that we can provide more webinars. I don't see any questions right now in the chat, but I don't know if anybody has any questions that they want to raise their hand, and maybe, we're a small group, maybe you can ask the question if you want, or we can do that as well. Oh, here we go. Ellen has a question for Lara. It says, what was the name of the machine you used for texture?
At the beginning, I used the Tax Plus, and then I moved to the Zwickröll. Hi, Ellen.
Hi, Laura. Thank you. Sorry, I didn't catch the name. Zwickröll?
No, Zwickröll is a German machine.
Maybe Lara can type it in the chat box.
Yes. In any case, you can also find it on the paper.
Okay.
Thank
you.
I will write it for you.
I appreciate it. And hopefully it's a better price than the taxed machines that are so expensive.
It is more or less the the price is not really cheaper. The thing is a little bit more open for what is my experience. But in any case, really, they are quite similar.
Thank
Okay,
you.
we have another question here from Janine. What was the motivation for changing machines?
I have my labs and the fields in an external site just for berries, and the Tax Plus was at the main campus of the foundation. So when I got a chance to buy a new machine, I tried this one, the Zwickrol. And I thought it was important to have 2 different machines to compare eventually. And at the same time, as I said, I was quite attracted by the fact that it was more user-friendly, I would say. And so this is why I chose this machine as a second choice in any case.
Ara, I want to just complement that question because, going through the process of deciding what we were going to use for the VacCAP at the beginning, this is 4 years ago, we looked into some alternative option, at least for the US market. We noted that the Tax Plus has very good tech support. I don't know in Italy if that's the case, but that was one thing that we found that the tech support was very good at that time. And then the Zwickrol, yeah, that was the downside of the Zwickrol here in the US. So if you look into those options, think about the logistic of getting support for the machine, that play a role in your decision. That's why then we end up using the Tax Plus, and also we had the machine already on site.
Yeah, I agree.
Do we have any more questions for our speakers? If you want to unmute yourself and ask the question, that you can also do that. Fernando, I think that you can go ahead and unmute yourself if you want to ask the question.
Hello, everybody. Yes, this question is for Dr. Iorizzo. So you mentioned there that with the smaller size berry, you have increased water loss. And if you want, I guess I'm trying to think about it in my head. If you wanted to, when I think of shelf stability, and please correct me if I'm wrong here, like when I've tested blueberries and tested them for shelf life stability, what I do is I notice that they dehydrate a lot quickly, a lot faster, generally speaking. Well, they dehydrate at some rate, right? And then if you want to improve shelf life, you would try to stop the dehydration, which means that then you would breed for a larger berry. But then can you clarify for me the link between the bigger the berry and the texture? Because if they're both kind of headed in the same direction, then it almost seems like you would just increase size overall just to improve texture.
Yeah, Fernando, thank you for the question. So first of all, between size and texture, there is no correlation. I mean, the correlations between the size measured at weight or height of the berry, which are highly correlated between each other, was very no or minimal correlation with all the texture parameters, okay? So then, so there is no real contribution of size to texture. Relative to the weight loss, the larger the berry, the less the water loss, to the point where after 2 grams, we know that this threshold, this plateau, this relationship plateau at 2 grams, we did across 3 studies, and we know that weight loss really plateau at after 2 grams, independently of the size. But small berry have definitely significant higher water loss but also increase the wrinkle in the lower berry compared to the larger berry. I hope that answers your
Yes, no,
question.
I see what you're saying. You're saying that if you have so there's like, just to repeat, just so that I understand here, you said that if you have there's no correlation between the size and the texture,
No.
but then the losses in texture, they sort of cap out at 2 grams, even
The
how
loss
do
of
I explain
water.
it? Yeah, I guess.
The loss of
Yeah,
water.
the loss of water
Yeah.
the loss of water and its effect on texture is minimized at 2 grams. Is that
No.
what you
So
mentioned?
yeah, there is no relationship between water loss. We correlated water loss measured because we measured the individual berry at 2 time points, right? There is no correlation between water loss and the texture parameters. It seems that the water loss really doesn't have a big role on affecting the mechanical texture. Yes, you can see 0.3, 0.2 correlation, but like not 0.7, 0.8, or the water loss seems affecting the appearance of the wrinkles, okay, but not necessarily the change mechanical texture.
Okay, gotcha. Thank you so much for that.
Thank you.
Great. We have another question here from Jennifer. Says, Massimo, you mentioned scar size not being correlated with texture. Was there any correlation between scar size and storage in your work?
Yeah, not that we could see, no. And we evaluated a large number of samples. And yeah, that's based on our observation. And I don't know if Lara in the breeding program observed something different, but not in our data sets.
No, I agree. No correlation for storage.
Yeah, I want to just say one thing, just to make sure. So sometimes a breeder evaluate wet scar. What we measured is the size of the scar. We did not evaluate wet or dry scar because it was very, very time-consuming process and very objective, subjective evaluation. So you want to still evaluate the wetness of the scar. Yes, you can add that in the macro of your texture analyzer or the pipeline that you build. We could have added wet scar as one of the parameters to measure, but we decided not to do it. But what we measured is the size of the scar.
Is there an association between scar size and weight that might have an impact on water loss?
Not that we have noted, Jim. Thank you for the question. No.
Okay. That's a good thing.
Yes, yeah. It seems that, like, I mean, I think that what we are finding is that, chemistry doesn't play a big role into, we measured sugar, not only bricks, we measured sugar using near, which is very good predictive of the individual sugar. We measured organic acids, we measured the bricks, pH, tartarial acidity. Seems that they are independent of the texture. And then we measured these, the size, the scar diameter, and seems again be relatively independent of the texture. So really, the way to select for texture is measuring the texture. And what we see is that it's not one component as you measure with the Firmatech that measure only one parameter. It seems that multiple components contribute to texture. So it seems that for doing, if you want to invest really on selecting for texture, you need to start to use this method that dissect texture more than what one of the other method does.
But that's really great that essentially the chemistry of taste is separate from texture. So
Yes.
they can be selected independently of each other.
Yes, that's a good thing. Yes, exactly.
Thank you.
There's one more question in the chat from Janine, who asked, do we know which texture parameters correlate with human preferences?
You know, Lara may have some other data, maybe, but the previous, yes, we have the previous webinar by Hilko. You can go to our webinar page where we basically did the study on relating texture, the same texture parameter that we presented today to sensorial texture, and there is one parameter, maximum force, which is one of the parameters that we indicate as the key for selection, that it's the best predictor of sensorial texture, in particular of springiness, hardiness, and crispness, which are key for getting a very firm berry. And then there is another study that it's about to be completed where with consumer panel, not trained panelists, which is indicating that at the increase of young modulus, which one of the parameters that we suggested for selecting, it's correlated, positive correlation with the increasing in willingness to pay by consumers.
I put there in the chat again where you can find all of the webinars, the recording of all of the webinars, including the one that Massimo was just mentioning. You can just go to our YouTube channel and you can find all the recordings there, including this one is going to be posted there maybe in the next couple of days. Well, we're a little bit past the hour, and I don't see that there's any more questions.
Yeah. Lara, did you add something else?
Yeah, I just wanted to add something to the question about the texture parameters that correlate with human preference, and I think I'm super lucky that I can taste wonderful tastes of blueberries, and actually this is exactly what gave me the input also to work on texture more than 10 years ago. And the good thing is that I mean, we for sure one thing is science, and what we are doing correlating specifically liking and the specific parameters, but then for consumers as well, and for producing a new variety and a new product, let's say. This is exactly what it tells you, that it can be different. I mean, you can have a firm berry, and I mean, in a multi-component way, firm with a different juice, and juice can also be measured, juiciness somehow from the size of the cells. You can have crunch. You can have a lot of different connotations that may bring to the consumer something good. And in the end, what is good is that we have a lot of options to work with breeding, and that I mean, you can be really happy when the grower has an easier life working with this berry, and gains more, and the consumer likes it more. So I'm pretty sure that in the future we will be able not only to call but also to produce much more diversity than what we have now for the consumers.
Maya, sorry,
Yes.
1 second. I want to just add the last note. So some preliminary data. Why it's important to start to understand this phenotypic method for phenotyping texture? It's also because some of the preliminary data we are getting on the genetics of texture are indicating that texture is complex trait, like in other crops, other small fruit, other crops. So then probably for the next for some time we will still be need to evaluate texture using this phenotypic method, because it doesn't seem to be easy to do single marker association selection for texture, at least so far.
Right.
Thank you.
Well, it seems like there's no more questions, and I really want to thank Massimo and Lara for fantastic presentation and sharing all the research and wonderful information with all of us. And for everybody that attended, thank you for attending. And as I said, the link to the recordings is in the chat. You can find our previous webinars there as well. Well, with that, I think we're going to conclude the webinar of today. Thank you very much to everybody.
Thank you.
Thank you. Thanks a lot.